to our next speaker, who is Christophe Avenel from the BIIF facility, which is part of the Swedish NMI Node. So please, Christophe, go ahead.
Yes, thank you. Can you hear me well?
Yes, perfect.
Good. Thanks for the introduction. So yes, I'm Christophe Avenel. I'm working in Uppsala, Sweden, as part of the bioimaging informatics facility, which is itself is part of the NMI node of Euro-BioImaging. As the bioimaging informatics facility, we provide support mainly on image analysis, support training. We also develop tools and give support for these tools for both image data analysis and also visualization of data. And today I will talk a bit more about this aspect of visualization. Unfortunately, the user is in parental leave, so he will not be able to join. So I will do my best to present also the biology side of the project. So excuse me if I say anything wrong about that, but I will try my best. So let me see now. Yes, this is our typical kind of projects in the node. We mainly work on microscopy, 3D light sheet data, things like that, where we do image analysis. But we also work on spatial omics data, visualization, pre-processing, and analysis, and also quality control. And that's what I will show you today. So the project we'll talk about has actually been published recently, last week in Cell. So you can check the paper now. And it's about cellular architecture of neuroinflammatory lesions and multiple sclerosis pathology. So it's a collaboration between multiple groups, including Mats Nilsson group in Stockholm and also Gonzalo Castelo Branco lab, where each group bring their own knowledge. And we use the facility here for like how to share and display, visualize, and explore the data mainly. So I will show you a bit more about that. But first, to give you a bit of context and understand the background, the idea here was that neuroinflammatory diseases like multiple sclerosis are really complex and quite challenging to study. So in this project, the user explored the dynamics of these neuroinflammation by using a mouse model. And the idea of the mouse model was that if you administer a peptide to the mouse, then it induces experimental autoimmune encephalomyelitis, EAE, here on the left. And that kind of replicates the main hallmarks of multiple sclerosis. So then using this model, you can study like both the temporal and the spatial dynamics of the disease progression. And then they used human data also next to that to correlate the findings and check if they find something interesting. To do all of these, they use data that is called in situ sequencing data here, extracted both from the mice and from the human using similar technologies. And these in situ sequencing were extracted at multiple time points for the mice, so from early to late stage, and also a different zone of the mice from I will come back to that, but from the lumbar up to the brain, to see how the disease progresses. But first, the main question is, what is this in situ sequencing, I guess? So I will explain that very shortly, just so that you understand the main idea, but I will not go into details about the technology itself. But just very shortly, in situ sequencing is a method for targeted analysis of RNA molecules. And this in morphologically preserved cells and tissue sections. So basically, you get the information of the RNA and DNA genes targeted analysis directly on the tissue with spatial coordinates and with subcellular resolution. So you can get for each one of your cells the information of which genes are present inside the cell. This is using fluorescence cycles and different techniques to get the different genes inside the cells. So once you have this, you can do cell segmentation and then affect to your cells each genes inside. And then you can do, you can like find the spots in the cells. From these cells with genes inside, you can do like clustering. So you can get cell information, like a cell population of what you have. So you can get different clusters, and then you can annotate these clusters based on the differential gene expression inside each cluster. So depending on the main gene inside, or like the list of main genes inside each one of your cluster, and with experts that know what these cells are supposed to be, you can annotate your population. And then, as this is spatial information, you can plot that back on the spatial tissue. So you can get all your cells with their specific type annotated more or less precisely. And this for all the section you exported. So this is what was done here in this project. And this was done in many different sections and at different time points. So the main idea here was that you can see then with these annotations how the disease and how the inflammatory response evolves. So basically, they exported, they extracted in situ sequencing in 4 different sections in the mice: lumbar, thoracic, cervical, and brain, and also in 4 time points: pre-symptomatic, symptomatic, the peak disease, and the late stage, 30 days after. Then they also observed the symptoms during these different stages, like limb weakness, tail paralysis, and so on, so that they could correlate the symptoms with the disease evolution. And then they could like focus on understand how the inflammatory response evolves over time in the different mice. But also, as we have the spatial information inside each of the in situ sequencing data sets, we can try to understand the spatial dynamics. So they were annotating also regions of interest based on the cell populations in each local regions. And then they utilized these computational framework to understand the progression inside each section. And then also from early to the more complex stages where the disease was inside of the different sections. And this was to give potential implications for like targeted treatments. In parallel to that, they had also similar in situ sequencing done on human samples with multiple sclerosis. So that they could correlate the findings in the mice model to real human data. This was done with the 10x genomics platform called Xenium that is doing basically in situ sequencing, but at a larger scale, and with the automatic machine to get all from the beginning to the endpoint. Using this, they were able to identify some genes of interest, especially SERPINA3 as a gene, as a key modulator for the inflammation, both in the mice and in the human samples. Yes, and they were also able to like do the same computational framework to find the pathological annotations of the different sections and so on. I will show you that a bit on the data later. But as part of the facility, what we offer is also we offer a visualization tool that is developed both in the lab, which we collaborate with, and in the facility, that is called Tissue Maps. And this is a viewer for spatial omics data based on, it's a web browser viewer. So you have a web version, and you can also install it locally on your desktop, or use it in a Jupyter notebook. And the idea is that it's GPU accelerated, and you can do quality assessment, you can visualize your data, you can do processing, you have some plugins and so on for large scale omics and digital data. So here, what you can load in this Tissue Maps tool is both your images, So any layers that you have, it can be like multiplex data or just DAPI. In this case, it was like DAPI images as background of the in situ sequencing. You can also load regions of interest if the data was annotated by an expert, or if you will have like cell segmentation or similar. But more specifically, you can load markers, and you can load millions of markers on top of your data. So here we were able to load all of the cells that were classified in the in situ sequencing and display some annotations for each one of these cells. This can be loaded directly from ANDATA object, which is the object that is created in Python when you do spatial omics, like spatial transcriptomics and so on. So you can directly load that in tissue maps. And so for this project, we shared online as part of the publication tissue maps web interface that you can check on this link here. I can put that in the chat if you want later, sorry. Or like if someone can put it in the chat, then you can explore the data, and I will give you a small demo of the platform now. So the platform, you open it, you will get into a web page where you have access to all of the tissue samples that were extracted. So you have like the 4 different time points from very early without any symptoms to the very late 30 days later. And you have the different sections that were analyzed from the lumbar here in the early stage, all the way to the thoracic, cervical, and brain sections of the mice. That were only analyzed for the peak and the late stage, because the disease progresses from the lumbar all the way to the brain. So it's like to see how it progresses. And here on the right, you have access to all of the data for each one of the cells. So you can like zoom in and see down to the single cell annotations or like scale. And then for each one of these, you can select the data you want to display on the right, going from the metadata, so like the sample ID, the area of the cells, and so on, or like the quality control. So if you select, for example, the total counts of per cell, then you will get how many genes were expressed for each cell. So you can click on a particular cell and see how many genes were found in this particular cell. And then you have all of the annotated data, going from very broad data with like about 10 different cell types that were annotated, all the way to very high precise annotations with about 40 different cell types more precisely. And you can see how they are like expressed in different regions of your data. And if you click on one, you can get the cell type of this one. Then you also have like the spatial neighbors graphs that you can display. So basically how the cells were connected together to do a neighborhood interaction and so on. So then you get like this kind of graphical representation of your data, and you get that also for all your samples. You also have access to the compartments that were annotated by pathologists, depending on the cell composition, cell population of the different compartments, going from the cortex and so on in the different regions that were used to find the genes of interest. And finally, you have access to like the list of all the genes in your data. So you can select one gene of interest, like MEG3, for example, and see where this specific gene is expressed at each time point and each section. I can also shortly mention that we have plugins. So if you go in the top section here, you can select some plugins of interest. For example, you can display the feature space corresponding to your data here on the right. So you can see how the UMAP was computed from these different genes inside each cell. So for example, if you select one part here, you can see on the left side where this specific region is expressed in your data. Thank you. That was it for me.
Perfect. Like I saw that it was 3 and you were like, thank you. So with that, I hope
Try to keep it to 15 minutes, which was difficult with all that biology.
yeah, and thank you for having the live demo. That is absolutely fantastic. And with this, maybe we can take one quick question. If I see any enthusiasts with their hands up. If that's not the case, then maybe I will ask a quick one, Christophe. So I see that a lot of the things that Tissue Maps is using is based on standards, which are rather, let's say, flexible right now. So you're using AND data and things like that, and they're not necessarily very stable. So have you had issues with it, or are you working closely with the developers, or how's that working for you?
No, it's tricky to follow the changes in the format world, especially now we're trying to add like ZAR also, because the community is moving from AND data to more like spatial data, which is using a different format in the background. So yeah, it's a permanent work on maintenance and trying to follow the community, but I think it's worth it so that users can load their data and visualize it super simply and fast. Yeah.